Welcome to our lecture on the cardiovascular system, talking specifically about the heart. In this lecture, we're going to learn about all of the major components of the cardiovascular system, focusing specifically on the anatomy and the physiology of the heart. We're going to discuss the cardiac cycle in depth from multiple different perspectives, and we'll discuss many different factors that can influence the heart's rate and contractile forces. Just to orient you, the heart is located in the mediastinum of the body. So here's a transverse plane, and you're actually looking upward right now. So here is the heart. If you're looking anterior and posterior, here's the heart. It's going to be located right in between the lungs. So here's the right lung and the left lung. This region here is the pericardial cavity, so that's going to surround the heart. Also surrounding the lungs, we're going to have the series of different muscles and the ribs, of course. In the back, we're going to have the vertebrae and the vertebral column. Some major things we should be looking at here, some structures. Here's the aorta of the heart. We also have the pulmonary trunk, which is going to be headed out towards the lungs, right? And again, that pericardial cavity. And we'll look at more specifically at the overview of the heart as we go forward. So this is what we're looking at if we're looking directly into the mediastinum. Here's the superior vena cava, which is going to be the returning blood supply into the heart. Here's the aorta, which is going to be the blood supply headed out. And every time that you see something red, it's going to be oxygenated blood, and blue is going to be deoxygenated blood. If we're looking at the heart itself, here's the apex. We have an inferior surface, this is the left surface here. Again, it's going to be in between the lungs. Here's the right and the left lungs. The pleural section has actually been cut here to reveal the lungs, but the lungs are also in their own cavity. That's that pleural chamber. See, and then again, this pulmonary trunk going to go out to the lungs. I think I've touched on everything here. So the heart is actually held in place by this sac. It's a pericardial sac. Forms this pericardial cavity here. It's basically, if you can picture putting your fist into a lightly inflated balloon, you can see that you would end up with this region here that would kind of be like a balloon shape around the heart, and that's to allow the heart to contract, so it's going to have much less friction with the surrounding environment. We have two different sides to the pericardium. The parietal layer is going to be the external side. It's going to be interacting directly with like the lungs, for example, and the visceral side is going to be interacting directly with the heart itself. Again, this region in the center is called the pericardial cavity. So the pericardium has two major layers, the visceral region and the parietal region. It has an outer section and an inner section. So we have an outer section that's going to be fibrous and very strong, and then an inner section which is serous. It has some liquid in there that's going to allow for the contractility with less friction. So the visceral and the parietal layers are separated by that serous cavity, which has that fluid in the middle. And the heart itself has three layers. So the epicardium is this region here. Right, so the pericardium is the pericardial sac that we just talked about. The next layer is going to be the epicardium. Then as we head internally, we have the myocardium and the endocardium. And again here, the pericardium lining the wall of the heart itself. The first thing that you're going to reach here, if you do the pericardium, we're going to have the visceral layer here of the serous pericardium. That's going to touch the epicardium. And then we are going to have the myocardium here. This is the actual heart muscle, the cardiac muscle. Then here we have some vessels. The heart itself has its own blood supply, and we'll talk about coronary circulation a little bit later on. And it also is going to have this region here. These are called trabeculae cardiae, and this is so that the heart has contractility. So it's going to be able to stretch and contract and stretch and contract. We have four major chambers of the heart. So we have two upper chambers, those are called atria, and two lower chambers, those are called ventricles. Now the atria are going to have a little bit of a thinner wall than the ventricles, because the ventricles are actually going to have a lot more contractility force, and also the left ventricle is going to be much thicker than the right ventricle. This is because the right ventricle is going to lead into the lungs, which is going to have a little bit less resistance than the contractility it's going to have to take from the left ventricle to send blood out through the aorta to be able to supply the rest of the body. But I digress, so let's talk about the figure that we have in front of us. So the top of the heart has this aortic arch. That aortic arch is depicted as red, of course. It's going to have oxygenated blood, and it's going to have three major trunks that come off of here. This is the brachiocephalic trunk, the left common carotid artery, and the left subclavian artery, and they're going to go exactly where you would expect them to go. The brachiocephalic is going to head into the brachia region, and cephalic being head region. Common carotid is going to go up, run along the carotid. Subclavian is going to run underneath the clavicle. Coming in We're going to have the vena cava. So this is going to be bringing in deoxygenated blood. Again, that's depicted as blue, and you're going to be able to see that throughout the rest of this lecture. Everything red is going to be sending oxygen out. Everything depicted as blue is going to be deoxygenated and coming in. Let's see. So here are the ventricles. Here's our left ventricle. We also have the left atria. Up here on the right side, we have the right atria, and then the right ventricle down here at the bottom. You'll notice several different things on the outside of the heart here. So these are coronary arteries. These coronary arteries are going to be the blood supply that supplies the cardiac tissue itself. We also are going to have some serious fat around here, so you'll see a lot of this these fat sections here shown in, well, yellow. Let's see what else we have here. Descending aorta. What else? Oh, and these are the regions that are going to be going out to be getting blood, sending the blood out to the lungs to be able to get oxygen. So this here is the left pulmonary trunk. It's going to be headed out, and then it's going to be coming back in, oxygenated. It's the pulmonary trunk coming this way. These are pulmonary veins. On the right side, we have the same thing. We have the right pulmonary artery, and we also have the right pulmonary veins. All right, so the pulmonary trunk is going to get split into the pulmonary arteries, and they're going to be going to either side, the left and the right, and then returning blood supply is going to be coming in through the pulmonary veins, and it's going to be headed back into the heart itself. All right, let's see. So the chambers of the heart have the two upper atria, again, the two lower ventricles. We talked about that. It's just a different depiction of it. Here's the arch of the aorta, as we talked about, with the three major sections coming off, the subclavian, the carotid, and the brachiocephalic. Coming in here, we have the superior vena cava. This is going to be deoxygenated blood returning in. We also are going to have the pulmonary arteries and the pulmonary veins. This gives you a better view of it, and I want to just take a second here to tell you that in general, when we talk about circulation, when we talk about oxygenated blood, we are talking about arteries. And when we talk about deoxygenated blood, we're talking about veins. The only time that we're going to see that switched is when we are talking about blood that is coming out from the heart and going to the lungs to be able to return back to the heart. So in this case, the arteries are still going to be having deoxygenated blood. So this is the only time in the entire body that you're going to see arteries depicted as blue. Usually arteries are depicted as red. And no, it's not an error. It's because these arteries are actually, for this moment, deoxygenated. They're going to go into the lungs. They're going to return oxygenated. This is the only time that you're going to see veins that are depicted as pink, right? They're deoxygenated as they head out to the lungs and then return as oxygenated in the veins, and then that's going to go into the heart and then go back out through circulation. This is going to return into the left atria, by the way. All right, so from the left atria, then it's going to go into the left ventricle and get ejected out through the aorta. All right, so that's just kind of an overview of everything that we want to be seeing about the heart. So then let's talk about the flow of blood. So the blood's going to head into the right atrium from the vena cava. We have the superior and the inferior vena cava, and also the coronary sinus. So don't forget the fact that the heart itself has its own blood supply. That's called coronary circulation, and the heart tissue takes up so much energy to be constantly beating and uses a lot of ATP. It's also going to run through a lot of oxygen very quickly. So after the heart, after the coronary circulation goes through and feeds the heart, it's actually going to feed back into the general circulation. So it's going to feed back into the right atrium through what's called the coronary sinus. So anyway, the coronary sinus and the superior and inferior vena cava feed into that right atria. All of that is deoxygenated blood that comes in here. Right atrium. Then it's going to go through this tricuspid valve into the right ventricle. Okay, that right ventricle is going to contract and send it out through a pulmonary valve, which is then going to send it out into the pulmonary trunk and send it out through these pulmonary arteries. Again, these are blue. Only time we're going to see blue arteries out into the lungs, where they're going to get oxygenated. We'll talk about that in a respiratory chapter, so we'll gloss over that for now, but then they're going to come back with oxygen inside that blood supply, and it's going to head back into the left atrium, go through the bicuspid valve, this is also known as the mitral valve, and then head into this left ventricle. And as I mentioned previously, this left ventricle is much, much thicker than the right ventricle, and again, that's because the right ventricle only has to contract to send it out to pulmonary circulation, right, out to the lungs and then back in. But this left ventricle is going to have to send it out through the aorta and out to the rest of the body. So that's going to take a lot stronger force of contraction. Additionally, I want to take a moment to say that this arch of the aorta is very important. The aortic arch is going to be able to contract and expand, so it's going to be very elastic. And that's going to allow it to be able to absorb a large high-pressure flow, and then also to be able to withstand that moment of dub when we're going to have a lack of flow. Additionally, there's these receptors. There's a ton of receptors in the arch of the aorta, but that's going to check things like blood pressure, etc., and that's going to be something that the body's going to respond to in terms of maintenance of homeostasis for your blood supply. Okay, so this is what I just showed you in the diagram, but now we're looking at an actual animal, right? So here we have the atria here. It's going to be cut open. That's going to, I'm sorry, the atria here going to be cut open. It's going to head through this tricuspid valve into the right ventricle, and then it's going to go out to pulmonary circulation through the pulmonary trunk, return back in to the left atria, go to the left ventricle. Again, it's going to do that through that bicuspid valve or the mitral valve, and then that's going to get head out through the arch of the aorta to be distributed throughout the body. All right, so some of the other things that I failed to talk about when I was talking about the blood flow or a lot of the connective tissue. So let me go over a lot of external things in this diagram. So some things here, for example, like this ligamentum arteriosum is going to connect the aorta here down to the pulmonary artery. Fun fact, that actually used to be a part of the blood supply in the developing fetus. So developing fetus actually has 4 arches that come out of the aorta, and one of them is going to end up turning into this ligamentum arteriosum, and in the adult we only end up with 3. Let's see, what else did I not talk about previously? We talked about, did we talk about valves? We did a little bit, but not a lot. So let's talk about valves. All right, so we have 2 different sets of valves here. The tricuspid and the bicuspid valve are going to work in concert to let blood in from the left and right atria into the left and right ventricle. Right, so they're going to be open at the same time, closed at the same time. We also are going to have pulmonary valves, right, and that's going to allow blood to go out into the arteries. And at the same time, we're going to have, so these are called like semilunar valves, right? And we also are going to have the valve that's going to extend out of the atria of the, out of the left ventricle into the aorta. Right? And those 2 are going to be open at the same time. So these guys are going to be open at the same time, and then those 2 are going to be open at the same time. And in between, we have a moment when all of the valves are closed. And what is the purpose of the valves? Well, the valves are there to regulate the blood flow to make sure that we don't get backflow from these chambers into those chambers. Right? So it's a directional valve. Blood goes this way to that way only. It never goes this way to that way unless there is some sort of pathological condition. Once it goes from here to here, then it goes out through this valve. Again, this is a one-way valve as well, that direction. So nothing is going to allow any backflow. And this is going to be important because we're going to have pressure that builds up that then has to get released, and that's what's going to translate into what we call our heartbeat, right? What else didn't I speak about here? These chorda tendente we didn't talk about. Those are going to be responsible for holding the different regions of the valve in place, so kind of allowing them to remain stationary as everything beats around them. We also have a lot of muscle here. So this is the papillary muscle. We'll talk about how that works and how that fires, but that's going to fire in concert from different nodes where electricity is going to jump from one point to another to allow the contraction of the heart. All right, I think I touched on everything in this image. So let's move on. So on the left side of the heart, we are taking in oxygenated blood, so it's coming back in from what's called pulmonary circulation, so back in from the lungs, and then it's going to go from the atria into the ventricle, which is then going to contract and send blood all over the entire body, right out through the aorta. And because we need much more pressure to be able to go out to the aorta than we do to be able to go into the lungs, the left ventricle is going to be much thicker than the right ventricle. So it's going to be pretty immediately clear which side of the heart you're looking at when you're doing a heart dissection when we get to the laboratory, because the left ventricle is going to be drastically larger than the right ventricle. All right, some other things that we didn't really talk about but are going to be super vital in heart function is going to be the fibrous skeleton. So this is going to be all of the scaffolding, right, the foundation, where all of the regions attach. So where the heart valves attach, where the cardiac muscle bundles attach, it's going to be involved in preventing the heart valves from overstretching. So it's going to be kind of a stop factor or limiting factor. And last but not least, but very importantly, it's an electrical insulator. So remember that we're sending electrical impulses from one section of the heart to the next, And so this is going to act like the wrapping around your electrical cord to keep the electricity from traveling from one cord to another. All right. So actually, let me step back. So we were just talking about heart valves. So let's show the heart valves here before we move forward. So this is going to be a transverse plane of the heart where we're just going right underneath the aorta. Here we have the pulmonary valve, and you can always tell the directionality of the valves. This is actually going to be headed, say, towards the screen. These guys are going to be headed like away from the screen, out through your computer. And you can tell because of the way that these are going to open in one direction. So this is only going to open towards us. And then it's going to, if you were to picture that you were pushed forward on this valve here, it would fill with blood and therefore prevent it from opening, right? It doesn't open that direction. And so the backflow itself actually serves as the pressure like a reservoir to help prevent the opening of the valve. This here is the pulmonary valve, so this is going to head out into pulmonary circulation. This is the aortic valve. This is going to head out through the aortic arch. These two are going to open at the same time. This one here is the bicuspid valve, and this one is the tricuspid valve. These guys are actually inside the heart. They're going to be the difference between the atria and the ventricle, and they are also going to fire at the same time. So these two, all four closed. These two open, all four closed. These two open, all four closed. These two open, all four closed. So there's a moment in between where all four of the valves are closed, which is going to make sure that we have no backflow in between opening the other set of valves, right? So you can picture this like turning on one faucet and then turning it all the way off before you turn on the other faucet to make sure that you don't have the mixing of the water. All right, so as I just mentioned, the point of the right and left atrioventricular valves, these are going to be the regions that are going to allow blood to flow from the atria into the ventricles, but specifically prevent the backflow from the ventricles into the atria. Again, all of this is a one-way system. We also have the right and semilunar valves. These are going to prevent the backflow from the arteries into the ventricles. All right. This is what a valve is going to look like. So when a valve is open, it's going to allow blood flow, right? When a valve is closed, it's going to, the back pressure is going to bounce off of this valve and not allow any blood to go this direction, and also at that time no blood's flowing this direction either. So when the valve is closed, nothing's headed one way or the other. And that's going to be partially responsible to these chordae tendente. When they are slack, they're going to allow things through, right? At the same time, we're also going to have blood pressure this direction. So we're going to allow the blood pressure to go from high to low in this moment in time. But if this pressure is higher than that pressure, it's going to close. So we're not going to allow backflow. And at that point, we're going to have the contraction or taut chordae tendente. Here's another section of the heart here. Here's the valves that we were just seeing in the diagram, but now we're looking at them in an organism. So here's the bicuspid valve, right? The mitral valve. Come on, mouse. Here's the bicuspid valve and the tricuspid valve. Again, these guys are going to be open in concert. You have the pulmonary valve and the aortic valve, and you can see they're headed the same direction. And they're also going to be open in concert because they're going to be pushing blood both out to the lungs and out to the body at the same time. So the ventricles are contracting at the same time. This is going to be an up-close view of the aortic valve, and it's a superior view. You know that you're looking down on it because these guys are going to actually be moving inward, so they can come out of your computer screen but won't be able to go. So if you could picture that you were pushing pressure into your screen, it wouldn't allow you, but pressure from the back of the screen would allow those valves to open. All three of those would open. And this region here is called the cusp, right? It's a semilunar cusp of the aortic valve. This whole thing is called the aortic valve. Now, when one set of valves is open, the other set is closed. And that's important because we want to make sure that blood is flowing in one direction. We don't want backflow to ever happen. And all of this happens by pressure, and so you want to make sure that it's going in one direction. We have pressure from high to low, and then if we have pressure from the other direction, it's not going to allow that to happen. And we'll talk about how that happens throughout the entire cycle when we look at things like the EKG. Again, here's our pulmonary valve. Here's a aortic valve. These guys are both going to allow blood to come out from the ventricles. So from the right ventricle, blood's going to go to the pulmonary valve at the same time as the left ventricle is going to be sending blood out through the aortic valve into circulation. On the opposite side of that beat, we're going to have the bicuspid valve and the tricuspid valve open to allow blood to flow from the atria into the ventricles. All right, and at this view, we've actually removed the atria, so we're actually looking right in between. We're looking down into the ventricles, so the atria would be kind of in front of your computer screen. If we look from the opposite side, here we have, the tricuspid valve, the opposite side, the opposite part of the beat. So during this beat, these are open and those are closed. During this part of the beat, these are closed and those are open. And just to be clear, there is a fraction of a moment in between where all four of them are closed, because the opposite option would be that there would be a fraction of a moment when all four would be open and we couldn't allow blood flow to have any backflow. All right, so that's what it looks like in the valve. So let's talk about systemic circulation and pulmonary circulation. We're also going to hear later on about coronary circulation. So lots of different types of circulation. And circulation just means systems of blood flow, right? So when you see things that are red, it's going to be oxygen rich. When you see things that are blue, it means oxygen poor. And let's just start here with 1, from 10 to 1. So 10 would be the very end of the cycle, meaning the blood is coming in from the vena cava. It's going to head from the vena cava. Again, it's connected from the superior vena cava, the inferior vena cava, and it's also coming from the coronary sinus, so from that coronary circulation that we haven't quite talked about yet. Going to come in here, to the right atria. And from that right atria, it's then going to head here into the right ventricle. From the right ventricle, it heads out to go to the lungs. Going to come back in, right? So that's going to head to the lungs here. We'll talk about respiration in an entirely different chapter. Then it comes back in, heads into, from both sections, from both the left and the right side, into the left atria, then down into the left ventricle. Contraction of the left ventricle ejects blood out through the aorta into systemic circulation, which means the rest of the body circulation. So pulmonary circulation would be circulation within the blood, within the lungs. Systemic circulation would be circulation of the rest of the body, and coronary circulation would be circulation specifically for the heart tissue itself. All right, so here's what's actually happening when we talk about all of the different regions. I talked about the different steps. Basically what happens is from 3 different sections of deoxygenated blood are going to pool. So we have coronary sinus blood, which is coming again from the heart muscle itself, the heart tissue itself. Then we have the inferior vena cava and the superior vena cava, which are going to be drainage of deoxygenated blood from other regions of the body. They head into the right atrium. Then they're going to go into the right ventricle. Again, they're going to do that through that tricuspid valve right here, and that's going to be when we have high pressure here and low pressure here. So high pressure here is going to then pop right on through to here, and then when this part is contracting, so we have high pressure here, these are going to be closed. So it's a one-way valve, right? All right, so it's coming in through the tricuspid valve into the right ventricle. Right ventricle is going to contract. Again, when it contracts, it cannot go backwards through this valve, so it has to go forwards through that valve. That valve is the pulmonary valve. Right? From the pulmonary valve, it heads out into the pulmonary trunk here, into the pulmonary arteries. Again, this is the only time that you're ever going to see arteries depicted as blue. So when it seems kind of confusing, you just picture it's still deoxygenated. It's heading to the lungs, and it doesn't have its oxygen yet. Once it has its oxygen, it's returning, which makes it that it's going to be coming back in as pulmonary veins, which is, again, confusing because every other time you see veins, they're going to be depicted as blue. Usually veins are carrying deoxygenated blood. Anyway, these pulmonary veins that are oxygenated are going to come back in here. This is number 6 through the left atria. From the left atria going through that bicuspid valve here to the left ventricle. When that left ventricle contracts, again, this one is closed during contraction, so it's only going to be able to go out through this one here. Again, that is the aortic valve. Through the aortic valve, it's going to head out, this is 8 through the aorta, and then out to systemic capillaries at the head and the upper limb through those 3 major branches of the aorta. I know that's a lot. Don't worry, we're going to go over it several times as we head through the rest of this lecture. All right, so let's talk about coronary circulation. So this is the 3rd type of circulation that we were talking about previously. And basically what this is circulation that's going to feed into the heart tissue itself. So the blood is going to flow through these coronary arteries, and that's going to deliver oxygenated blood just like it does for the rest of the body. But again, the myocardium is going to be very, very active, constantly using ATP, so it's going to be high energy dependent. That means it's going to be kind of stealing the oxygen very quickly. And so if we were to use regular circulation, we would end up with less oxygen available to this tissue. So the coronary circulation actually is going to feed directly off of the oxygenated blood that comes out of the heart itself. It doesn't go out into circulation. It goes directly around the heart and then back in through the right atria. All right. So these coronary veins, again, that does the same thing that veins do in the rest of your body. They're going to be removing carbon dioxide and waste from that myocardium region, and they have these different converging regions, and these are called sinuses, and the major one of that is going to be the coronary sinus, and the coronary sinus is what's going to lead back into that right atria with the deoxygenated blood to make sure that we get oxygen back into that blood before it goes into circulation. So this is what coronary circulation looks like. So where does it come from? So directly off of the ascending aorta, so directly with that freshly oxygenated blood that just came out of that left ventricle, we have this coronary artery. We have 2 of them. We have a left and a right, and they're going to feed opposite sides. The right coronary is going to actually head down here and has this major branch called the marginal branch, and then feed down into the region that is going to be the right ventricle region. We also are going to have this left coronary artery. It has a circumflex branch and an anterior interventricular branch, and then that's going to feed into the left ventricle. And if you look on the opposite side of that, so it's depicted as clear, obviously it's not clear, but just so you can see what happens here, so this branch here is going to wrap around and become the posterior interventricular branch, and that's going to feed into the back of the ventricles themselves. In terms of the coronary veins, it's a little bit simpler. This here is the great cardiac vein. And then we also have the anterior and the middle cardiac vein. The middle cardiac vein is in the back here, again depicted as clear but not actually clear, wraps around here, and all of that meets into this region here called the coronary sinus. So again, that's in the back. So the great cardiac vein wraps around. We have the middle cardiac vein here wrapping up, and then the small cardiac vein and the anterior cardiac vein, all of that meeting here in the coronary sinus, which again is going to feed directly into that right atria to head right back into the blood supply to get oxygenated. Okay, so this is what cardiac muscle tissue looks like. Cardiac muscle tissue is very specialized because it has to be both contractile and very responsive to electrical impulses, and also going to be using a whole lot of ATP. So these are these particular cardiac muscle fibers is one cell. You can see that it has this nucleus here, and it has a ton of mitochondria. Why does it have a ton of mitochondria? Because it needs to use a lot of ATP, and so it's going to need to have a lot of mitochondria to make that ATP. This here is the sarcolemma. We have these desmosomes that are going to be, I don't know if you've ever talked about desmosomes before, but desmosomes are a way by which cells are going to adhere to one another. And so desmosomes are going to mean, because we have just a couple of them in between, we're going to have the ability to stretch as opposed to something like a tight junction, which would be like kind of sealing it very tightly. These are going to allow for contraction and contractility. And these here are specialized regions. They're made of what's called connexons. It's not important, but they make these gap junctions. So gap junctions are going to be another way of connecting cells, but instead of the way these desmosomes are just kind of anchoring, these are going to be for information. So it's going to be picking up information, like electrical impulses are also responsive to things like epinephrine, norepinephrine, through these gap junctions. So in this way, these cells are able to actually talk to one another, kind of the way you would string a tin can from one house to another. Also, in between our cells, we have what's called an intercalated disc. So these intercalated discs are depicted here, and they actually are held together by these desmosomes that we were talking about. Let's see, what else am I missing? I think we're clear there. Okay, so this is the overview of a cardiac muscle fiber, and there's a lot of things to take a look at here. So just to orient you, you've probably seen some of these things before. This region here surrounding it is called the sarcolemma. That's depicted here in blue. The nucleus of the cell is depicted in purple. Surrounding that, we have the mitochondria. Those are going to be where we're creating our ATP, of course, right? And then we have these filaments. We have thick and thin filaments. So thin filaments are made of actin. Thick filaments are made of myosin, and as you can kind of imagine, if you were to pull these two together, we can actually get to the point where these thick filaments are kind of touching one another, and the thin filaments, because you see the thin filaments don't run all the way this way, and the thick filaments don't run all the way that way, so this can move over that way, and this can move over that way, and that's what contraction basically does. And to measure these contractions, we look at many different things. This whole thing is called an I band, right? From one section to the other, and that's one sarcomere. The Z disc is from one of your intercalated discs to the other of your intercalated discs, right? And the M line is going to be the region that's basically going to show your contractility. So how far can this go before it touches that? And those are those thin filaments we just talked about. Actin, again, they don't go all the way through. If you follow them, they stop here. They could contract all the way to here, but at the same time, remember, this one will be coming that way. So how much contractility room do we actually have? And that's depicted here by this H zone, and the M line is the region where they would meet, so the median line. All right, so I think we've touched on everything in here. Oh, this here is a sarcoplasmic reticulum. It's going to be a region around the cells that are going to be kind of like a connecting region, so we can discuss, information is going to be able to pass from one section to another in that region. Okay, so the neat thing about cardiac muscle is that they are autorhythmic, and that means that they can self-excite. That means the left cell excites the right cell, excites the right cell, kind of like a wave passing through a stadium. And it's actually really neat. You can see this even in culture. You can induce them to beat, and you can change their beating rhythm. If you just have a look at culture of cardiac tissue, you can make it beat by adding beat faster or slower by adding different things like epinephrine, etc. And the way that this works basically is that we're going to create these action potentials, and they're going to be auto-inducing. So we create action potential and then create another action potential, and over and over and over. And that triggers these contractions in the heart. And there's a ton of different cells involved. These cells form what's called a conduction system, and the conduction system is basically the route that the action potentials take throughout the heart muscle. This here on the bottom is something that you're going to see a couple more times throughout the slide. So let's discuss what's happening here. Basically, we have what's called a membrane potential, and it's measured in negative or positive millivolts. And what happens is we have a change, a flux of ions, and the ions are going to be positive ions that come in, and that's going to cause an increase or positivity, right? So it's going to go from negative up to positive. And once we reach a certain threshold, we cross that certain threshold, it's kind of the point of no return. Everything floods in, increase the millivolts, everything floods out, and then we have a moment of kind of a refraction. And that action potential is going to be passed along, so it moves kind of like a wave throughout the tissue itself. So let's talk about the conduction system. So the conduction system, if you picture kind of like a wave, it actually has more like a train station, right? So we have one place that is going to be the origin. This is where all of the information starts. That's that sinoatrial node. This is going to be the first point of contractility and the first point of electrical excitement, and then it passes information off. So there's like a little bit of a jump here from the sinoatrial node to the atrioventricular node here, and also over to the left atrium. So the atrioventricular node here, the AV, is going to be connected to what's called a bundle of His. And that's depicted here. If you look at this whole thing kind of in red, is the electrical conduction, I guess. Basically, that pathway is going to go through that bundle of His until it reaches these here bottom things called Purkinje fibers. These Purkinje fibers are contractile, they're electrically excitable, and they're going to be responsible for, they're not contractile, they're in association with contractile tissue, they are electrically excitable and then excite the contractile tissue directly next to it and cause the contractions of the heart itself. Some other things you may recognize here, these valves, right? And then this is going to be these other valves, right? These valves are going to go from the right atria into the right ventricle. This one goes from the left atria into the left ventricle, right? And then out from the left ventricle through the aorta, right? So it's the aortic valve out into blood supply, just to kind of reorient you as we're going through here. So what's neat about the sinoatrial node is that it's autorhythmic, and that means that they make these natural pace. So the pacemaker of the heart is this region that's constantly going to initiate action potentials at a steady rate. And it's a steady rate until it receives signals from external sources. So for example, something like epinephrine or adrenaline can cause the heart rate to increase. So if you picture anything that is the parasympathetic or the sympathetic autonomic nervous system, that is going to affect the heart rates. But they themselves don't set the fundamental rhythm. And fun fact, they do have a maximum amount of contractility that they can induce. So we are going to reach a maximum heart rate, and then that's going to stop there, because if we got any further past that maximum heart rate, we can induce something like cardiac arrest. All right, let's go back to that action potential that we talked about now that we're previously talking about these charges. So this is called membrane potential. It's measured in millivolts, so it's measured in electrical charges, and this is negative and this is positive. So if there's a region where we're going to be heading into a positive when we were normally, up until that point, negative. And again, I mentioned that's because we have a positive influx. So we have an inflow of sodium ions, and sodium ions, as you may know, are positive. We have these specialized channels. They're called voltage-gated fast sodium channels. And basically, they open, and all of a sudden, there's a flood of sodium. As this sodium floods in, as you can imagine, we have positive charge added, so the negative charge is going to be negated and eventually overwhelmed. So now we have a positive charge during that time frame. That's considered our plateau region. We're going to maintain that depolarization. This whole thing means depolarization. Basically, as soon as you go from one, this is negative, one charge, to the other, here it's positive, that's called depolarization. So now we have these depolarization wave that's going to be maintained in the plateau state, and that's going to happen because calcium inflow. So first we have a rapid influx of sodium, which is positive, and then we're going to have an influx of calcium. Again, it's positive, but it's a different ion, and that's a special type of voltage-gated channel. So we have two voltage-gated channels. One is for sodium, and it's rapid. It's fast. Instantaneously, we have a ton of sodium flooding into the system. And then we have a slow, sustained plateau, which is maintained because we have calcium coming in through those slow-gated calcium channels. And then slowly, we're also going to have outflow of potassium at the same time. Okay? Now, when those potassium channels open, and now we have potassium outflow, now we're going to start to see what's called a repolarization, so bringing it back down to a negative charge. And repolarization happens because we close those calcium channels, so the calcium's not going to be coming in anymore, but the sodium is still going out, so we're going to have an outflow of sodium when we have voltage-gated sodium channels opening. So that's our third voltage-gated channel. All right, so positive ions in very quickly from the sodium channel. Positive ions in sustained on a more slow basis through these slow calcium channels, and then positive charge negated or eliminated back down to the negative charge when the potassium channels open and we have an efflux or potassium going out. That whole thing is one action potential period. So we have the depolarization and the repolarization. Additionally, we can look at other things like refractory period, which is the time that it takes for the whole thing to settle back. So basically, as soon as this process starts, we're here, we can no longer initiate a response. We've already initiated a response. We're not able to initiate a response, not even during our depolarization event either, or sorry, our repolarization event, I apologize, until we end up back to where we started. So this is that refractory period, and until we end up in here, we're not able to actually induce another action potential. And during this part here, which is as soon as we pass over our threshold and get into the positive section to about here, again, we're over that threshold, and until we hit here, then we're out of that contractile period. So this section here is basically when we have induced a positive charge, where we previously had a negative charge, we had a negative membrane potential. Now we have positive membrane potential, we are seeing contractility. So, as I mentioned, this costs so much energy. Cardiac muscle has to constantly create ATP. It does it on a steady basis. And it does that by anaerobic cellular respiration and also something called creatine phosphate. So we have a very short energy burst that we can get from creatine phosphate. So let's talk about this one first. Basically what happens is, let me talk about ATP if you're not familiar with ATP. If you were there for my Krebs cycle lecture, you're going to tell me, oh man, I already heard all of this and I apologize, but I'll simplify it. So ATP is an energy carrier molecule. It's a triphosphate form. It's a higher energy version of a molecule than ADP, which is the diphosphate. Basically, ADP has 2 phosphates, ATP has 3 phosphates. Higher, lower energy. And when ATP releases that phosphate, energy is released to be able to make this lower energy product, right? And when that energy is released, it drives other reactions. So anytime that you see it kind of with this funky, that's going to indicate that it's high energy, and a nice smooth is going to be the lower energy. So creatine is going to have a lower energy and a higher energy form as well, right? Creatine phosphate. So basically ATP releases its third phosphate to make ADP and free phosphate, which releases energy. That energy is going to be picked up by creatine to make creatine phosphate. That creatine phosphate is going to release its extra phosphate to be picked up by an ADP molecule over here to be able to have a high energy ATP to form a reaction that's going to allow muscle contraction when it releases its free phosphate. And that's going to be a really quick duration of energy. It's going to be about 15 seconds. And basically creatine phosphate is going to serve as like an intermediary between relaxed and contracting muscle to be able to allow the contraction to occur very quickly. But creatine phosphate is going to get depleted in about 15 seconds. So a much longer way of getting energy, so the duration of energy for this is about 2 minutes, is to be able to take glucose and perform glycolysis. Now, as you may remember when we talked about the Krebs cycle, anaerobic respiration is actually much less efficient than aerobic respiration. So for glycolysis, we're going to be breaking down a 6-carbon glucose into 2 3-carbon molecules. Those are the pyruvic acids. Don't worry, I won't quiz you on that. But basically, we're going from 1,6 to 2,3. And this 1,6-carbon molecule is going to release 2 ATP when it's chopped into 2 3-carbon molecules. Then that pyruvic acid has to get converted, because normally that pyruvic acid is going to enter into what we call cellular respiration pathways in the mitochondria, and that's going to create all the rest of the ATP that we normally get under aerobic conditions. But in anaerobic conditions, pyruvic acid is going to get converted to lactic acid and get converted and depleted out into the blood. And so that's going to be how the ATP is going to arise from anaerobic glycolysis. Let's talk about an EKG. So I'm going to give you a second to orient yourself. There's a lot of information that can be read from this diagram. And an EKG is basically measuring the electrical changes that are going to happen throughout each heartbeat. Now, as we talked previously, we have 2 major types of contraction events, right? We have the atrial contraction, where we're contracting both atria, and that's going to send blood through that tri and bicuspid valve into the ventricles. And then we have ventricular contraction, whereby we're going to contract the ventricles and go out into either pulmonary circulation or systemic circulation through the lunar valves and the aortic valve. So that's also going to be depicted in this diagram here. So atrial contraction here is depicted here in blue, ventricular contraction in yellow. So again, the first thing that has to happen, contraction of the atria. Second, contraction of the ventricles. So what are we looking at here? So this is looking at the millivolts. Remember, we talked about an action potential and how an action potential could be sustained with electrical, or is an electrical impulse that is sustained basically. In order for that to happen, we have to go positive when we were previously negative. Well, here we're looking at the millivolts of the actual heart tissue itself. And so this interval here, nothing's actually happening in this interval here. From this here, we have a P wave, and that's called the P to Q interval. And this is basically going to be the initiation of the atrial contraction. So the P wave starts, and that starts the contraction of the atria. Then atrial contraction is finished, and we're going to enter into the Q to T interval. Right, this QT interval here is going to be the very start of ventricular contraction, and then obviously we're going to finish off with some electrical impulse as well. So here we have this Q to R is going to be that spike. QRS is that spike that you typically see when you look at the little monitor and you see the green line. And then the S to T section is going to be the second section where we are still having ventricular contraction occurring. Then after this, we have the T section, which is going to be kind of our refractory period. And don't worry, I know I kind of glossed over it. We're going to go over in a little bit more detail in just a minute. All right, some new words are being introduced here. The word diastole means relaxed, and systole means contracted. Okay, and if you've ever had your blood pressure taken with a blood pressure cuff, and we're going to do that in lab as well, then you'll start realizing what we're looking at is actually the pressure change between the relaxed ventricles and the contracted ventricles. That's the difference between systole and diastole blood pressure, which is your XYZ over ABC. And so basically what you're looking at is the difference between when the heart is contracting and when the heart is relaxing, or when the ventricles, I guess, are relaxing and contraction. All right, so let's start here with one. The first thing that has to happen is we have to have the contractile fibers producing that P wave that's going to start the contraction, right? And as that P wave happens, now we have contraction here in that SA node. Again, we're overlaying several pieces of information. We talked about the electrical impulses starting at the SA node and then jumping over to the AV node, following out through the bundle of His, also jumping over to the left ventricle, right? So from the SA node, it goes from the right ventricle to the left ventricle. Those contractions happen at the same, from the left atria to the left, let me back up. The sinoatrial node is found in the right atria, and it is going to send information from the right atria to the left atria so that these guys can contract at the same time. It also is going to send information to the atrioventricular node, which just a moment later is going to start ventricular contraction, and that's going to head out through the bundle of His down into the Purkinje fibers and start contraction. Okay, so that's the secondary part when we're talking about ventricular contraction. Right now we're talking about atrial contraction, so atrial systole. So the P wave starts. That's going to start the initiation at that SA node, which then also goes over to the left atria to start contraction. So the atria are going to enter into systole or contraction during the P wave. After that, we have the depolarization of the ventricles, which is then going to lead to contraction of the ventricles, right? And that's going to produce that QRS complex, so that spike, QRS here. After that, we end up with ventricular contraction, and that starts almost immediately after in that S wave that's here. That goes from S into T. During T, we have the repolarization of the contractile fibers, so everything is kind of having a reset, right? We have a refractory period, and we end up with relaxation, and then we're going to start this whole thing over again. So first we have to initiate the contraction of the atria, boom, then we have to initiate and have the contraction of the ventricles, and then we have a relaxation period during the T wave where we have repolarization of the ventricles, right? So one cardiac cycle is going to be the contraction and relaxation of both atria, which is that first section, right? Again, that happens by the sinoatrial node. The sinoatrial node is going to send information from the right atria where it's located to the left atria, so they're going to contract together. Then they're going to relax, that's diastole. Remember, during the contraction event, we're going to be opening up those valves that are going to be connecting the atria to the ventricle, right? Namely the tri- and bicuspid valves. Now, during relaxation of the atria, now we're going to have higher pressure in the ventricles, and so those valves are going to be closed to prevent backflow. And then we're going to have systole, right, contraction of both ventricles, and then diastole of both ventricles. And there's a lot of things that are going to change throughout here. So the electrical events we've just kind of talked about. We also have a lot of pressure changes, and I've touched on them as we talked through the different steps of the blood flow, but we'll talk about them specifically and how they're related to that EKG. We'll also talk about the heart sounds, like the lub and the dub. We won't harp on that too much, but you basically can hear the valves opening and closing when you go to the physician and they put the stethoscope on your back as you breathe, for example, or on your heart, I'm sorry. We also have volumetric changes that are happening. Obviously, as blood is filling the chambers, they're getting larger and more volume. As they're flowing out of the chambers, they're getting smaller with less volume. And then, of course, the mechanical events, which is the contraction itself. All right, so I've already kind of gone over the electrical events. So here's that EKG. The first thing that happens is that P wave. That P wave induces the atria to contract. So that's atria systole. That is going to lead all the way through the beginning part of the QR and then into the S. In the middle of the RS, we're going to transfer from contraction of the atria. Now we're going to have a second where we have atria relaxing. The ventricles are going to enter into systole between the end of R to S all the way through T. And then during T, we're having kind of a refractory period, a relaxation period. And then we'll enter into the next cardiac cycle. Don't worry, we're going to overlay all of these, and I'm going to go into them independently as well. So let's talk about the pressure changes. There's several different things on here. So this is the aortic pressure. So if we're looking at what happens inside the aorta, it's going to be completely dependent on when the left ventricle contracts, right? So you can see that the aortic pressure and the left ventricle pressure are going to be very similar just for this contractile period, right, during ventricle systole. We're also here looking at the left ventricle pressure throughout the entire section. That's going to be, again, depicted in blue. Here we have the left atrial pressure. That's going to be depicted here in green. Okay, so several different things that we're looking at. Pressure is going to be depicted here, again, low to high. It's in millimeters of mercury, but basically what's going to happen is that initially we're going to have low pressure here in the left atria, right, and the aortic pressure is going to remain about the same during that time frame. So we have low pressure in the atria, also low pressure here in the ventricles. And then as we enter into the contractile period, we're going to end up with a burst of pressure in the atria. Again, the atria don't end up with the same amount of pressure increase as, for example, the ventricles do, because they don't have as much. They're just pushing the blood from the atria into the ventricles. They're not pushing the blood into, for example, pulmonary or systemic circulation. But after we push the blood out of the atria into the ventricles, right, that these valves are going to close, that bicuspid valve is going to close. After that closes, now we end up at the same time, if this is happening to our electrical impulses, are going to induce us into ventricular systole. So at this moment, bicuspid valve closes, a heartbeat later, the ventricles enter into systole, which means they contract. When they contract, they increase in pressure until that aortic valve opens. When that aortic valve opens, both the aorta and the left ventricle are going to increase in the pressure until the ventricle systole relaxes. At that point, when we end up with less pressure in the left ventricle, the aortic valve is going to close. So again, we don't want backflow to occur back into the ventricles. And then we have a secondary wave. It's called a dichrotic wave, where the aortic valve, because of the fact that it just closed off, is going to have a quick little boost in pressure, and then aortic pressure is going to fall back down to where it was previously. The left ventricle pressure is going to drop very dramatically because it's already pushed everything out into the aorta, and now it's also going to be starting to open, so all of a sudden it has a lot less blood in a larger space. And then here, the bicuspid valve is going to be open again, and the left atria is going to, the left atrial pressure is going to end up remaining low. And then we're going to just lather, rinse, repeat here as we enter into that next section. Okay, this I'm going to kind of gloss over, but these are the heart sounds that you're going to hear. We have 4 different heart sounds, so lub, dub, right? And that is going to hear as we enter into systole for the atria. So contraction of the atria is going to allow the opening of the valve. So really what you're hearing is not the contraction of the heart itself, but the opening and the closing of the valves. And so when you have the valves open and close, well, in between atrial and ventricle systole, you have them closed, right? Because you don't want backflow. So when the atria are contracting, it's going one direction. Oh, it's all going the same direction, but they're going to be, the tricuspid and the bicuspid are going to be open, and then when we enter into ventricular systole, the tricuspid and bicuspid are going to be closed, but we're going to be opening up the aortic and the semilunars, and that's here. And then as we're closing them again, that's in this relaxation period, you're going to be able to hear the closing of the valve. So when you're actually listening to the heart, that lub, dub, what you're hearing are the valves themselves. And what people are listening for are arrhythmias, so if it's going to be beating in an odd rhythm, for example, or something that sounds kind of sloshy, like a heart murmur might occur if you have a bad rhythm, or you might end up with the ability to hear something like a valve defect, where you could actually hear liquid flowing backwards during this section, for example, when this should just be the pressure of the valve closing. You might be able to hear a leaky noise going the opposite direction from ventricular into the atrial regions. Oh, sorry, I'm lost for a second. All right, here's the volume changes. So we were just looking at the pressure changes, and the pressure changes are going to be related to the volume changes. So kind of keep that in mind. And again, we're going to show a slide where they all overlay at the same time. So the volume changes are going to be shown here in the ventricle itself. So once we enter into atrial systole, the ventricle volume is going to increase a little bit, right? Because the atria are going to be pushing the blood into it, so it's going to be stretching. Then as we have that moment where we close the valves, now we're going to enter into ventricle systole, so we're going to be squeezing the ventricles. The volume of the ventricle is going to change dramatically. And this region here between fully open and fully contracted is called stroke volume. And this is one of the things that they measure when people might have issues with cardiac function is their stroke volume. So it's basically the end diastolic volume over the end systolic volume. And that's going to indicate how much you're actually pushing out of your ventricle per stroke of the heart, right, per heartbeat. And then as we enter into the relaxation period, the ventricle is going to slowly end up filling back up to where it was normally, and then it's going to enter into, again, another atrial systole, where it's going to have it increase because, again, the atria are contracting, forcing more blood into the ventricles. So that's what's going on with the ventricular volume. And these are the mechanical events. So these are things that you should actually already kind of understand because we have talked about them extensively throughout this lecture, but just to overview you, the phases of the cardiac cycle are going to be first atrial contraction. Again, what happens here, the atria are going to be contracting, pushing blood into the ventricles. You can automatically tell left and right because here the left is going to be thicker, the right is going to be thinner. And also this is bringing in deoxygenated blood, and this one is bringing in oxygenated blood. After the atrial contraction, we have a moment of isovolumetric contraction, and this is important. This is a moment when all of the valves are closed, right? And then at that moment, we're going to have the ventricular ejection. Immediately after that, we have the ventricles contracting. That's going to send the blood out through, this here is going to be going into pulmonary circulation. This is going into systemic circulation, right? So we're going to be going through the aortic valves and the semilunar valves respectively, and then it's going to be coming back in. That's called isovolumetric relaxation. During this time, we're going to be pulling the blood back in through the, from the right atria is going to be receiving from the vena cava, the left atria is going to be receiving from the pulmonary veins, and then that's going to lead to the section where we have ventricular filling, right? Ventricular filling, again, is going to come through that section here. It's going to be kind of a slow, leaky pull as the, back up. These guys are going to be completely contracted here, these ventricles. They're completely contracted, they push everything out, and then as they open back up again, slowly the blood is going to come back in, both into the atria and then leaking through those valves until it ends up here. That's going to cause isovolumetric relaxation. Eventually we're going to end up with equilibrium, so ventricular filling is going to occur. Then we're going to end up starting this whole thing over with atrial contraction, where we are pushing as much as we can into the ventricles. Again, that's here. And then isovolumetric contraction, again, where we have all four valves closed. There are two sections where we have all four valves closed, right? There's isovolumetric contraction section. Basically what happens is we're going to have the contraction of the ventricles pushing the blood out. But we want to make sure at the same time that we don't have any blood flow back into the atria. So these guys are going to close, and the semilunar valves and the aortic valve are going to close for just, or stay closed for that moment, right before these guys open. This slide looks like a lot, but if I were you, I would use this as a review slide. So this slide is everything that I've already told you kind of overlaid. So this is the EKG itself, so the electrical impulses. And the overview is all overlaid in the general idea of here's the atrial systole, here's the ventricular systole, and then this is the relaxation period. We also have the same thing I showed you in terms of the pressure for the ventricles, for the aorta, for the atria, and then also the heart sounds, again, lub dub, when all of the valves are going to be opening and closing. This is what happens for the ventricular volume. So as I mentioned, we have that increase where we have the push from the atria into the ventricle. And then we have the contraction of the ventricle, so we lose everything at that point. The ventricles are going to then relax. So we have isovolumetric relaxation, which is going to allow filling of the blood back into these until we reach the equilibrium. And then we'll get contraction event again, where we're going to be expelling the blood again from the atria into the ventricles. Just lather, rinse, repeat. Okay, so if I were you, I would start with the slide and kind of talk your way through, and anything where it felt like you didn't understand, then go back to the other sides or look it up in the textbook so that you have an idea of what you're missing. So, The blood that comes out of the heart is measured in terms of cardiac output. So cardiac output is a pretty simple definition of just the amount of blood, the volume of blood ejected from the ventricle, you can do it from the left or the right ventricle, into either the aorta or the pulmonary trunk each minute. So it's a pretty simple mathematical formula, and again, I'm not going to ask you to do anything super complicated in terms of the exam, but I want you to understand that we are looking at cardiac output in terms of the stroke volume, which is basically just the amount of blood that's going to come out of the ventricles in one beat, and we talked about that previously here, stroke volume, right? The difference between the end diastolic volume and the end systolic volume. How much we're actually able to eject from the ventricles, so that's stroke volume, times the amount of times that we do that per minute, so your heart rate, right? So the milliliters per beat times the beats per minute is going to give you the milliliters per minute, because we're just going to eliminate beats on bottom and beats on top. And that's going to give you your cardiac output in milliliters per minute. Now there are three factors that regulate stroke volume. We have preload, contractility, and afterload. And preload is basically what you put on it as you're about to contract. Contraction is the contractile load, and then the afterload is what's left after the contraction occurs. We have multiple different factors that are going to regulate our heart rate. And when you were present for our autonomic nervous system lecture, you'll recognize that almost everything that upregulates heart rate is going to be in the parasympathetic division. Is in the sympathetic division, and everything that decreases heart rate is in the parasympathetic division. So basically, fight or flight is going to increase your heart rate, and rest or digest is going to decrease your heart rate. I apologize that I said that wrong at the beginning of that section. And then hormones are also going to be involved in regulating your heart rate and cardiac output. The amount of ions, this is really, really important because as you saw, everything is influx of positive ions. So calcium levels, sodium levels, and potassium levels are all very, very important. In fact, if you end up with a deficiency of sodium, you can end up in cardiac arrest. Your age is going to regulate your heart rate, your gender, your physical fitness. So the more fit you are, the more responsive you are to different physical overloads. Also, your temperature can regulate your heart rate as well. So one of the first things that happens when you get really cold, for example, you're going to shiver, and then eventually you're going to end up not shivering at all, right? So your temperature can eventually end up making your heart rate super low if it drops very low. We also have a nervous system regulation of the heart, as just like everything. So I feel like every lecture we've talked about how the brain is going to regulate it. The brain regulates everything in your body. And we have a specific region of the heart that's going to be your brain. It's going to be the cardiovascular center that's going to regulate the heart. And that here is shown in purple. And that's going to have input that comes from the higher brain centers, like the cerebral cortex. Also from all your sensory receptors, so things like your proprioceptors. Where are you in relative to the rest of your body, for example, or your surroundings? Chemoreceptors, these monitor your blood chemistry. These are really important, and I know we've talked about them in terms of homeostatic conditions in other chapters, but if you guys are fresh to this, joining this lecture, chemoreceptors are going to be responsible for monitoring your homeostasis. In this case, talking about blood chemistry, but for almost everything in the body. We also have baroreceptors. These monitor blood pressure, and as you may recall, I talked about them being located inside the aortic arch. And so they're going to measure the blood pressure as the heart is contracting and pushing blood out, and also as it's relaxing. And so it's going to increase and decrease every cardiac cycle. Now, from the cardiovascular center, we took information in from the brain and from your proprioceptors, right? And then also, we're going to have information going out. That's going to go to two different ways. Now, as I mentioned, almost everything that's involving the heart is going to be upregulated by sympathetic and downregulated by parasympathetic. So we have cardiac accelerator nerves that are part of the sympathetic section, and these guys are going to increase the rate of heart contraction by increasing the spontaneous depolarization in the SA node, and then hence that is going to relate in the AV node. That's going to increase your heart rate. Now I want to have a little caveat here. There is a limit to how much we're going to be able to increase this, because eventually if we increase it too much, we end up with non-function. You still have to give the heart the entire time to go through its contractility cycle. And so if we were to allow this to end up completely unchecked, we can end up in cardiac arrest. So in the normal conditions, there's actually a regulatory stop point whereby we are no longer going to be responsive to an increase in this sympathetic pathway. But up until that point, it's going to cause an increase in the rate of the SA node firing, and therefore increasing the heart rate, and that's going to increase contractility of both the atria and the ventricles, also increasing stroke volume. So we're going to increase both stroke volume and heart rate. Both of those are factors in what? Cardiac output. So it's going to increase our cardiac output. Now that vagus nerve, it's going to be one of those cranial nerves. It's cranial nerve X. It's going to innervate the abdomen of the body, right, and it's actually going to be innervating the heart as well. And that's going to be involved in decreasing the rate of spontaneous depolarization, which is therefore going to decrease that heart rate. And as I mentioned, sympathetic and parasympathetic work in opposite directions, right? Rest and digest is opposite of fight and flight. And so it's not surprising to hear that this nerve is going to increase the rates of SA and AV node firing, while this nerve, which is part of the parasympathetic pathway, can decrease the rate. And so in this way, the cardiovascular center can increase or decrease the rate of cardiac output and contractility. Okay, so as I mentioned, cardiac output is comprised of two major things, stroke volume and heart rate, right? Multiply those two together, you get cardiac output. So how do we increase cardiac output? We can do it from two directions. All of these factors are going to increase stroke volume, resulting in increased cardiac output. All of these factors are going to increase heart rate, resulting in increased cardiac output. So again, we talked about preload, contractility, and afterload. And so now we're going to discuss that a little bit more. So increased preload is basically going to be the end diastolic volume. So at the end of the relaxation period, how stretched is the heart? And as you exercise, you end up with, so people who are more physically fit, end up with a increased volume inside the heart to begin with. So the heart's already going to be able to have the ability to contract more forcefully because it has an increased preload. We also have several different things that can be related to sympathetic stimulation of the autonomic nervous system. Some things like catecholamines, glucagon, which we talked about when we were talking about glucose synthesis and digestion, thyroid hormones we talked about during the endocrine system, and also the increase of calcium in the extracellular fluid. Remember, we talked about how calcium is super important in changing the membrane's action potential, or in creating that action potential by depolarizing the membrane. Again, that's going to cause an increase in contractility. And increasing contractility is going to increase the stroke volume as well. And last but not least, the afterload. If we have a decreased afterload, so that means that we have less in at the end. So if we have more in at the beginning, we contract more out, and we have less left at the end. All three of these are going to result in increased stroke volume. So how do we end up with a decreased afterload? We end up with that if the semilunar valves open a little bit sooner. And they open a little bit sooner if the blood pressure in the aorta and the pulmonary artery is lower. So that means if you have a lower blood pressure, then you can end up with a decreased afterload because more blood can go out, and that's going to increase the stroke volume as well. Basically less resistance that it's pushing against. All right, so we're going to switch to this side. We're talking about the heart rate. So the nervous system we've talked about extensively. It's going to be very involved in the parasympathetic and sympathetic nervous system are involved in cardiac output by increasing heart or decreasing the heart rates. So that's going to be regulated by the medulla oblongata. It gets information from things like the cerebral cortex, the proprioceptors, the baroreceptors, the chemoreceptors. We just talked about that, and that's going to result in an increased heart rate by having increased sympathetic stimulation or decreased parasympathetic stimulation, either one. Now, if we had the opposite, so if we had decreased sympathetic or increased parasympathetic, all of that would decrease the heart rate. So all of this is something that is able to be regulated, for example, when exercise stops or when the threat is eliminated, so you no longer have to face flight or fight conditions. Some other chemicals, several chemicals can help regulate your heart rate. Specifically, we talked about the extracellular calcium levels, but also catecholamine and thyroid. You might see that over here as well. So we can have that increase the contractility and also increase the heart rate as well by increasing the calcium availability, for example. Other factors that we spoke about, including your age. So infants and senior citizens are going to have a lower response, a lower ability to have a cardiac output. Also females, because opposed to males. People with lower physical fitness, so in poor physical condition, or in increased body temperature, or also in something like a low oxygen conditions. So all of these can affect your heart rate, which affects your cardiac output, and so does your stroke volume. Alright, so if we exercise regularly, it can do several things for our body. One, it can increase our cardiac output, as we just spoke about. Secondarily, it can increase your HDL, and you may remember from our nutrition and metabolism lecture that HDL is going to be the good cholesterol, so it's a ratio of HDL and LDL that's going to determine your cholesterol levels. So aerobic exercise can increase your HDL, which can end up making your heart healthier. Also can decrease your triglycerides. It also improves your lung function, because if your lungs are used more often, they have a better tidal volume and better respiratory capacity. Also can decrease your blood pressure. Again, that blood pressure is going to be responding to baroreceptors in the aortic arch. And so if you're used to regular routine exercise, you might not end up with increase in blood pressure immediately, so you can end up having better response to your stroke volume. Also, it can assist with your weight control, obviously, because aerobic respiration is going to be burning calories, so calories in, calories out. So if you increase the amount of exercise, you can assist the amount of weight that you're putting on. All right, so this part is going to be kind of, I don't know, it's fun for me. This is talking about different mechanisms by which we can help patients who have failing hearts. And as aspiring nurses, this is something that you may end up seeing later on in your career, depending on which career pathway you talk about. So this is showing a heart transplant. So this is talking about here, here's a recipient heart. So this is the original heart that's going to be removed and replaced. This here is the donor heart. And so we can do a partial transplant. We're basically just going to have the partial left atrium is going to be left, and then we're going to be putting on a new right atrium, for example. So if one atria isn't working and the other one is, or etc. So you can take a section of the heart and do it that way, or you can do a full heart transplant. And when we do a full heart transplant, we're going to suture basically all of the major sections together. So we're going to be suturing here the superior vena cava of the recipient to the donor's right atria, and on the bottom also the recipient's inferior vena cava to the donor's right atria. And then we're also going to be suturing the aorta down into the into the aortic arch, into the aorta. So all of these major vessels, well, the vena cava and the aortic arches all have to be sewn together. And now here's our fully transplanted heart with the sutures in all the right regions, and with any luck this will hold and eventually will work just perfectly fine for this individual. However, just take note, because it is a transplanted heart, although they do their best to make sure that they have matches for organ transplants, obviously in terms of like blood typing, etc., they're never going to be perfect, and so this individual is going to be need to take immunocompromisation drugs, so drugs that are going to help suppress, so immunosuppressants basically help suppress their immune system, so they're going to be able to not fight off this donor heart as non-self. This means that they're going to be immunocompromised because they are going to have lower amounts of antibodies, etc., almost intentionally. So they are going to be an individual that may need to pay attention to minor sicknesses, etc. All right, so here's some alternative cardiac assist procedures. So not everybody jumps immediately to the transplant. Obviously, transplants are going to be reserved for patients who have severe cardiac distress, and oftentimes there's very, very long waiting lists. So some options that we can assist, either as a alternate to a transplant or as a staving off device to kind of wait your time for the transplant, something called an intra-aortic balloon pump. So this is going to be basically a balloon that's going to be put on a catheter and put into your heart. And they're going to enter in through the femoral artery. They do this because it's one of the largest arteries in the body. So if you picture this as a series of road maps, this is going to be your major interstate. So they follow the major interstates until they can get into the heart itself. And then they're going to put that balloon wherever it needs to be. So wherever we have a problem with blockage, I'm going to inflate that balloon for you, right? And basically it's going to be coordinating with the ventricular diastole. And so this would happen if you had a hard time with the actual ability of the heart to pump. And so because it's going to be pumping in concert with the beating of the heart, it's going to be pushing the blood forward and backwards with the blood, with the pumping of the heart. So basically it's going to be inflated, pushing the blood both into the heart itself, which is going to improve the coronary blood flow. Oftentimes we have a major problem with the coronary circulation that's supplying the heart itself, so the heart is actually deprived of oxygen, so it's not functioning properly. So this is going to do two things. It's going to help improve the coronary blood flow and also push it forward through the peripheral tissue. So as the balloon is inflated here, it pushes the blood into the heart tissue itself and then also into systemic circulation. Then it's going to be rapidly deflated just at that ventricular systole, just before the ventricular systole, drawing the blood out of the left ventricle, so it's kind of sucking it in, and that's going to help make it easier for the left ventricle to eject blood. And it's going to be inflated in between heartbeats, so it's kind of a pulse, and that's called intra-aortic balloon counterpulsation. And so this is going to be something that's going to help assist with a heart that's not able to have enough cardiac output to be able to supply the needs of the body. This individual is probably going to be on the list for a transplant. We also have ventricular assist devices. Ventricular assist devices have been known to operate for many years without failure. So these guys could be an end stage, or they could also be an intermediate phase for someone who is awaiting the transplant. But basically, a ventricular assist device is going to be a mechanical pump. So it's going to be assisting the ventricles. Generally, they're used as a bridge to transplant, but sometimes it is a destination therapy. So sometimes it is itself going to be an interim solution. Perhaps if this individual is very old, right, in their 70s or 80s, and they might not be expected to have another couple of decades in them. So instead of waiting for a heart transplant that they might not get because they're going to be very low on the list, this might be something that might be their end stage destination therapy. And basically what happens is it can assist either ventricle, but the left is going to be most common simply because the left is the one that is going to be under the most pressure throughout your lifespan, is the most likely to fail. And basically what happens is going to help the left ventricle pump blood out into the aorta. We also have a right ventricular assist, which can help assist the right ventricle pumping the blood. Remember, that's going to go out into the pulmonary trunk, into pulmonary circulation. And in the most severe cases, we have a biventricular assist device that's going to help both of the ventricles, both the left and the right ventricles, perform their functions. And the type that these guys are actually going to need is going to depend on the patient themselves. So it depends on where they're going to have an issue. If they have failure of the left ventricle, they'll need the LVAD. If they have failure with the right ventricle, the RVAD. But it's very unusual to have failure of the right ventricle without failure of the left ventricle. Usually you end up with failure of the left ventricle, which then turns into failure of the right. So more than likely you need either an LVAD or a BVAD. What's interesting with these, though, is that these are going to be external. Well, I mean, this part is internal, but they're actually going to have a pump unit that's going to be on the outside of the body. So these guys are going to be carrying around with them sometimes an external control system on their waist or on a shoulder strap. They do have internal ones, but these are going to need to have batteries changed routinely, so it's going to be a kind of a cross between constantly undergoing a minor surgery or having something on the outside where you can change the batteries. So, all right. We also have something called a cardiomyoplasty, and basically what happens here is skeletal muscle is used to help assist the heart. And what they do is they'll take the left lat muscle, which is going to be the region where the heart's going to be around anyway. They're going to free it partially from the regions that connect it to the connective tissue attachments, and then wrap it around the heart, and then implant an electrical pacemaker so the skeletal muscle itself is going to contract in sync with the heartbeats. It's not going to contract entirely within sync with the heartbeats because we don't necessarily want to induce the pacemaker to change the heart signal. So we're going to end up with contraction about 10 to 20 times per minute, and several of them are going to fall in sync with the heartbeat, so hopefully it's going to be helpful. We also have a skeletal muscle assist device, which is similar to what they just talked about, except we're making a pouch that's going to be inserted between the heart and the aorta that helps as basically a booster heart, and it's the same thing. They stick in a pacemaker that's going to excite the motor neurons to make contraction occur in the skeletal muscle. So I've talked about most of these disorders throughout the lecture, but some disorders that we may encounter include coronary artery disease. I don't think I touched on this one. Basically, when we have plaques that build up, we can have atherosclerotic plaques that occur that can end up lodging inside the arteries of the heart. Basically, what happens if we have occlusions in the coronary arteries, then we're unable to get the perfusion of the cardiac tissue, so we can end up with necrotic tissue or fibrotic tissue in areas that are supposed to be contractile. And usually this is when people are going to end up getting a bypass. So we have 4 major coronary arteries, and we can end up with a quadruple bypass if we have to do all 4. Usually people get like a di or a tri-bypass. Atherosclerotic plaques build up within the body themselves generally, and sometimes we can what we call throw a plaque, right? So this is why they ask you to walk around when you're on long plane rides so you don't end up with something called deep vein thrombosis. And veins, which are found in your lower legs, can end up having clots build up in them if you're sitting with, say, your legs crossed or whatever. And then when you stand up and walk out of the airport, that clot gets dislodged by the sudden flow of blood. That's going to end up, or can end up, lodged in other places of your body. And if it ends up, for example, in your brain, then you can have a stroke. Depending on where it's located, you can end up with issues with, say, memory or motion function. We also can end up, or mental, we also can end up with something that could be a heart attack if that clot ends up lodged in one of the coronary arteries. Let's see, arrhythmia is when we're just going to have abnormal heart function or abnormal heart rhythm. I apologize. Instead of just lub dub, you might have a double lub or something like that. So this is something that would be detected, something like a heart murmur, detected at a normal physician's office, and something that you just generally keep an eye on, unless it's something that actually is going to interfere with your regular. I mean, it's an abnormal heart rhythm, but as long as the heart's working properly, they generally don't do much with it except monitor it. And then the last one here is a congestive heart failure, and that's what I talked about when we were going to need some of these ventricular assist devices, or that balloon that we talked about, or a full-on heart transplant. So that brings us to the end of our heart lecture. I really appreciate you sticking it out. You have a fantastic day.